If you are wondering why we didn't post a video or podcast last week, it's because our audio didn't record. So take 2. Yippee. Imagine moving to a different country by yourself at 18, getting a PhD by 26, and then decrypting the genome of one of the scariest viruses in the US and possibly the world at the time.
Flossie Wong Staal was a pioneering molecular biologist and virologist whose work reshaped our understanding of HIV and AIDS. Born in China and later making her mark in the United States, Flossie became the first scientist to clone HIV and map its genes, which was a breakthrough that opened the door to blood tests and life-saving treatments. Beyond her discoveries, she was also a role model for women and immigrants in science, proving that persistence and vision can break barriers. Today, we will be exploring her journey, her groundbreaking contributions, and the lasting impact she left on medicine and society.
Intro. Choo-choo, choo-choo-choo. So Flossie Wong Staal was born as Yi Ching Wong on August 27th, 1946 in, excuse my pronunciation, Guangzhou, China. Much is not known about her childhood or early life, but in 1952, after the Chinese Communist Revolution, wow, I really can't speak today, revolution, when she was 7, Flossie and her family fled to Hong Kong. There, she attended an all-girls Catholic school, and it is believed that here's where her love for science first bloomed. At the Catholic school, the nuns also asked Flossie to change her name to a more English-sounding one, and she decided on Flossie. So, like, her name wasn't actually Flossie. Her original name was Yi Ching. Actually, I don't know if... Was it... Because I know in some Chinese cultures it's, like, flipped. Like, the last name comes first. She changed her name to Flossie. And there's a cool story behind it, also. So, she was like, what should I change my name to? And she said... You know, there was that typhoon that just hit southern China. Let's do that. So she changed her name to a mass destructive typhoon.
Do you know, like, how destructive the typhoon was?
I did not look up how destructive the typhoon was.
Hopefully it was not destructive.
But I think it was. I don't know how destructive, but I fear it probably was destructive.
Dear. That's crazy.
At 18, Flossie ended up moving to the United States to obtain her bachelor's degree in bacteriology from the University of California at Los Angeles. She was the first woman in her family to receive higher education and graduated a year early cum laude with her bachelor's of science in 1968.
And bacteriology is such an interesting bachelor's major, like for undergrad. I guess because it's a different time.
I'm curious what the difference between molecular biology, microbiology, and bacteriology are. I mean, I guess I understand the difference between molecular biology and the other two, but right. I feel like microbiology and bacteriology are like very... I guess microbiology also includes like viruses and parasites.
Yeah.
And like, doesn't molecular biology also include that? You're the microbiologist.
Yeah, all I know is micro, not bacteriology.
Bacteria is in micro.
I mean the degree specifications.
Oh. Oopsies.
Anyway, so Flossie wasn't done there, i.e. changing her name and graduating with a bacteriology.
Bacteria. She graduated with a bacteria. I really want, you know, the giant microbes?
Yeah.
They make the little stuffed animals. My dad had an E. coli stuffed animal when I was younger, and when he was mad at me, he would take it away from me because I loved my E. coli.
My micro professor, my first micro class in college, she had the E. coli stuffy.
It wasn't my dad.
No, I didn't discover your dad's class until like last year. So, Flossie continued her doctoral education at UCLA, where she met Stephen P. Staal, who was going for his PhD in oncology, which is cancer research if you live under a rock. So, they got married in 1971 and eventually had 2 daughters, Stephanie and Carolina Vega Staal. Side note, so kind of hate that she hyphened her name, but her daughters just got their father's name, especially considering how important Flossie becomes. I don't know, like, I would be kind of upset if I didn't carry on my parents' legacy. Like, they only had their dad, who didn't really do anything. I mean, sure, he was an oncologist, but he didn't, like, discover important groundbreaking research. So, like, as, you know, the daughter of my mom, or hypothetically, if I were, like, Stephanie or Carolina, I would be you know what I mean. Anyways, so she graduated with her PhD in molecular biology in 1972. The following year, she became a postdoctoral fellow in the Robert Gallows Laboratory of Tumor Cell Biology at the National Cancer Institute in Bethesda, Maryland.
So close.
Oh god, it's so long. It's hard not to mess that up. But anyway, so side note number 2. There is speculation that Gallo is Carolina.
I don't know. Sometimes they say Caroline, sometimes they say Carolina, sometimes they just call her Vega, the second daughter.
Carol!
Carlos! Anytime I see Vega, I just think of Trina Vega from Victorious.
Yeah. Anyway, so, Mrs. Vega's father. So not sure how, if this is true, because the source that is definitely not maybe Wikipedia. But yeah, so we couldn't find the original article. You get the idea. So maybe not.
We could find the original article. We just couldn't read the original article because...
Is it not in English?
No, it you have to pay.
Oh, yeah.
I fear I wasn't going to pay for that.
No, if y'all sponsor us, maybe we'll pay for more in-depth sources. Anyway, yeah, so maybe Gallo is or is not her dad.
Instead of Stephen.
Regardless. Not relevant to Flossie. Well, it kind of is, but it's not. So Flossie's notable first achievement was regarding the human T lymphotrophic virus. Oh god. HTLV. Human T lymphotrophic virus. HTLV is a retrovirus, meaning that the genetic material of the virus is RNA when the virus is infecting the cell. The host cell will convert the viral RNA into DNA, which gets inserted back into the host cell DNA. So this causes the cell to make more viruses that are released throughout the body. HTLV, like many retroviruses, is spread through breastfeeding, sexual contact, and blood. It causes immunosuppression, making the infected individual susceptible to even more infections. So unfortunately, there is no true treatment for HTLV, so physicians aim to prevent the spread of disease by detecting it early.
Now, most researchers at the time did not believe that retroviruses could cause cancer. Well, Flossie provided definitive molecular evidence that HTLV was actually carcinogenic, aka causes cancer, and proved all of them wrong. Gallo even framed a letter from a reviewer who rejected their paper on the basis that it was a quote-unquote fact that human retroviruses weren't real. Like, I don't know, that's just kind of funny. He said, you say we're wrong? Stop calling me. Who's calling me? My mom? No! Anyways, I think it's really funny that he was just like, you're calling us out, we're actually gonna call you out. And then he put in a little picture frame. So he was like very proud of like Flossie, like saying strong in her beliefs and proving everyone wrong. She eventually rose to the rank of senior scientist, and then in 1982 became section chief for the National Cancer Institute Laboratory for Tumor Biology. Jesus Christ. It's said that around this time, Flossie co-authored more than 100 journal articles. For context, I've been trying to put out an article for probably 4 years. And there are some people who like pump them out really fast, but my PI does not put them out fast at all. But 100 journal articles is insane. And around this time, the AIDS epidemic started to become a thing. And by a thing, like, people were finding out that, like, AIDS was killing individuals. And most people have heard of, like, HIV and AIDS, and before we, like, actually go back to Flossie's stories, we're just gonna go through a quick run-through about the disease and its timeline and history, because there's a lot of misconceptions about HIV and AIDS, and it's really relevant to Flossie's research.
Yes. So, unfortunately, back in the 80s, during the HIV and AIDS crisis, people didn't exactly know much about HIV or AIDS, and this resulted in some very discriminatory beliefs. So, physicians noticed a pattern that HIV and AIDS were present in populations of gay men, hemophiliacs, which are individuals whose blood doesn't clot properly, heroin users, and Haitian people, which led to the association of HIV and AIDS with the 4 H's. The general population began to fear these people from these groups because they thought that just being around these individuals would lead them to become infected with HIV and AIDS. They didn't realize that anyone could get it if they were exposed to an infected bodily fluid. There's actually a story of a boy named Ryan White who contracted HIV through a blood transfusion when he started showing symptoms. And he was then barred from attending school because his parents were afraid, and other parents were afraid that Ryan would give their kids AIDS. So there are so many more instances similar to his. Like people refusing to shake somebody's hand just because they might be gay. So even though science and most people know better now, there's still stigma associated with HIV, AIDS, and gay men.
So what exactly is the difference between HIV and AIDS? HIV stands for Human Immunodeficiency Virus, and AIDS stands for Acquired Immunodeficiency Syndrome. Most people, including myself for a really long time, use these terms interchangeably, but they're not exactly the same thing. HIV is the virus that actually attacks the body's immune system, specifically CD4 lymphocytes, if you care. And this just makes it harder for someone to defend themselves against infections, illness, and cancer. And the dots should connect. But just because someone has HIV doesn't necessarily mean they have or will get AIDS. Even though there is no cure for HIV, there are now antiretroviral therapies, ART, which can help reduce the viral load, which is the amount of virus in your body. And with the help of ART, people who have HIV can now live longer and healthier lives. But in the 80s, they didn't have ART, and so people were left untreated for a really long time. When HIV is left untreated for about 8 to 10 years, that's when it develops into AIDS, which is the last stage of an HIV infection. An individual is considered to have AIDS when their CD4 cell count is less than 200 cells per millimeters cubed. So it's like a very specific cutoff, but that is once you have reached that point, that's where your body has no defense against anything, literally the common cold, any sort of illness infection could kill you. And it always bothers me that people act prejudiced against individuals that have HIV or AIDS, because those people, there's a common saying where animals are probably more scared of you than you are of them. It should be that way with people with AIDS. They are the ones who are susceptible to getting infected, to dying from whatever microbacteria that you just have living on your body. They could die from that. And so I feel like it's the responsibility of us to protect them if we can. But that's my rant. Anyways, back to Flossie.
So working with Gallo, who, little reminder, may or may not be her child's baby daddy, Flossie was the first person to clone HIV and determine the function of its genes. So this was a crucial step in proving that HIV was the cause of AIDS and to the eventual development of diagnostic tools and therapies. So furthermore, Flossie also discovered molecular evidence of microvariation in HIV, aka that HIV can make rapid, small-scale genetic mutations, which makes it harder to find and treat and yada 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 yada. So this means that it's evolving faster than medicine can keep up with, so just one medication will not be effective against it, which leads to the drug cocktails in the treatment of HIV and AIDS. So I think one of the coolest things about Flossie's work is the fact that she was making these discoveries so quickly, especially considering how new HIV and AIDS research was. So due to all of her hard work and incredible research, Flossie became the most cited female scientist in the 1980s. She had approximately 7,800 citations, which for the scientific community is a big deal. For anybody, that's a big deal. That's insane.
For context, my mom is a pretty good researcher. She pumps those papers out. I think she has 52. That's good. That's over the span of 20, 25 years. That's a good amount. Mm-hmm. So 7,800 is insane.
Because each citation is most likely another research paper written about this.
Yeah, exactly.
That's 7,800 new papers that stemmed from her research.
Right.
Insane. So Mr. Gallo said about Miss Flossie, she became a postdoc and she was the best I ever saw, before, during, or after. She was really sharp in things I needed to be sharper in. We talked every day. Yeah, every day, I can tell. Anyway, and that because of her, we became one of the most productive labs in the 20th century. So a few others in her lab agree that she was the reason the lab was so successful. People say that she was cheerful with a wonderful sense of humor and a flair for fashion. So a fun little tidbit, she also had a passion for competitive ballroom dancing.
So in 1990, that was a busy year for Flossie. She was recognized by get ready for some long names. She was recognized by the Institute for Scientific Information as the top woman scientist of the 80s. She left the NIH to become the Florence Seeley Rifford Chair in AIDS Research at the University of California, San Diego, and she divorced her husband.
Staal? That one?
Yes.
Okay.
Yes, that one. Wait. Yes, she did divorce that one. Don't worry, there's another one coming. But 4 years later, in 1994, she became the director of the newly created UCSD Center for AIDS Research and began investigating gene therapies for the treatment of HIV and AIDS. As if she wasn't doing enough already, at some point she married another guy named Jeffrey McKelvey, who was a neurologist, but she co-founded a biopharmaceutical company with him named Immusol. So don't know when she got married. At some point, she divorced that one and then got married to another one. But yeah, they created a whole company because she wasn't doing enough already. She had to do more. But in 2002, she retired from UCSD and became the chief scientific officer for Immusol, which she renamed iTherex Pharmaceuticals, which I low-key like Immusol better. Sorry,
too.
Queen.
That kind of makes me sad.
iTherex.
It's like
iTherex.
iPhone.
It's giving anthrax.
Yeah.
Not like anthrax
Immusol.
bacillus,
Just
but
because it has X in it. iTherex.
but like anthrax, like the company.
Oh yeah, there's one. Oh
Yeah,
my god.
yes, that one, the one that we pass all the time.
Isn't
Not
it
anthrax bacillus.
like the anthrax.
Yes.
Yeah.
Every single time we passed that, I was like, did they make anthrax? Because I didn't know what anthrax was for the longest time. That sounds so stupid. Especially considering my dad is who he is. I had no idea anthrax was bioweapon.
I didn't even know what it was. I thought it was a haircutting company because of the little scissors next to it. And then I met someone who worked at Anthrax, and I was like, oh. Dear.
I thought, okay, because I remember anthrax got spread by the mail, right? So I thought it was a powder that people put on the glue on the mail labels, and when you went to go lick it, that's how you got anthrax.
And the company did that?
No, I knew that those two were different, but oh wait, no, I definitely thought that anthrax made anthrax.
They're in business.
I wasn't old enough to know what the anthrax disaster was. Anyways, she made a company, and we don't like the name. The original name was cuter, and she refocused said company to treat hepatitis C.
It's still a great company. Our opinion of the name is irrelevant, truly.
Very irrelevant. We're just girls.
So Flossie mentions that she felt like she faced no discrimination on the basis of her sex, which for once, that's amazing. But it still stands that she was the first person to decipher HIV, and her work is still so relevant to today's evolving healthcare. Flossie was ambitious and stubborn in the best sense possible. Her first grant application to the UCSD was rejected because she was a molecular biologist who was trying to research in the field of immunology with no prior immunology experience. She didn't care that she didn't receive the grant. So Flossie did the study anyways, published the paper, and then sent the paper with her next grant application to the UCSD.
She stapled it, and she said, here you go. I did it anyways.
Also, here, babe, here's my experience now that I did it anyway.
Yeah.
So there's another account of her presenting her data at a conference where instead of using the traditional slides method that every other presenter used, she tried a brand new technology based on the recommendation of her daughter. You want to guess what that technology is?
Do tell.
It's PowerPoint.
Yup.
And then the next year, at the same conference, not a single person was using slides. They were using PowerPoint.
And when we say slides, I feel people are thinking Google Slides. I'm talking you put the projector in, it's old-timey
makes
slides.
A lot of clicky noise.
Yes, and it spins rotators.
I really like projectors. But anyway.
Projectors or PowerPoint?
Projectors.
Oh.
They make a nice noise, and
I like PowerPoint and my little animations.
Little shadow figures in front of the thing when you get bored. I don't know.
I am of the strong belief, though, that why do scientific PowerPoint presentations have to look so boring? Please, I understand that data is important, but the data can have pictures and look cute and have nice font.
Yeah, I'm not saying it has to be neon green. I'm not saying that's cute.
But
just
someone hire a director of marketing for PowerPoint presentations for
Research-
scientists.
Research does not have to be boring. Like, it's not boring, so why are we making it look boring?
Yeah. Like, the white slides with just the black... Like, at that point, just put a piece of paper up there and write your findings
And then
down.
you try to do anything else, and they're like, why would you do this? This is unprofessional.
I remember that I had for senior seminar class, I was so tired of the boring slides, and so I did I still kept it simple. I had a teal background and it was white, and I still used one of the respectable fonts. I think it was Garamond or something like that. My dad made me memorize all of the acceptable scientific fonts when I was younger.
Family of science over there.
Oh yeah, we keep viruses in our fridge.
Anthrax?
No, different ones.
I don't think that's even a virus.
It's not. It's a bacteria. But I had the nice PowerPoint and everything, and one of the comments that I got back was, "Make your PowerPoint more simple." And I did it. I made the next one neon.
Why'd you make it neon?
Because I felt like it. I felt jaded.
Out of spite.
Yes.
Yeah.
And I still presented it well, and I got a 100. Anyways, sorry, back to Flossie. It's been a long day and a long week, and this didn't record last week.
If we have to record this again, there might be tears.
Exploding. Back to Flossie. She passed away. I'm sorry, that's not supposed to be funny, but she did pass away from pneumonia on July 8th, 2020, at the age of 73 in San Diego. Now that I'm thinking about it, I'm wondering if it was COVID related. Because it's
In 2020?
2020, because
Yeah, 'cause
everything
it
got
was,
shut down for COVID
Yeah.
March 13th, 2020.
That's very possible.
And I remember when I was researching it, it said pneumonia with complications. And so
Yeah,
I'm
that was
wondering, well, just wait, because Flossie's research with all of her HIV related stuff actually laid a lot of the groundwork and framework for COVID research. And she's remembered by dozens of students that she mentored, many who became leading scientists in their field, her children, her siblings, and in a male-dominated field, she led with strength and kindness and intelligence and grace, and received so many awards and recognitions throughout her life. But it's still not super well known that her basis, her research basis was the basis for all HIV research. And again, her methods helped with with the COVID response in 2020. In 2002, she was named one of the 50 most important women in science, and in 2019, she was inducted into the Women's Hall of Fame. Her life is a testament of going after what you want and not backing down, no matter what people tell you. As a pioneer in virology, her achievement and intelligence live on, and every time someone is treated for a viral infection. And that is the story of Flossie Wong Staal. Yay, the end.
I have a question.
Yeah.
Did your dad know who Flossie was
I don't know.
before his...
I doubt it.
Probably not, but I don't know. I feel like...
I can call him. Momentary pause while we call my father, South Carolina's only virologist. Hello? Why is it not ringing? He's not gonna pick up. He hates me. This is the end of the video. It just cuts off with us FaceTiming my dad.
He called back and he said no. The end.